So, good evening, everyone. I'm Ananya Chatterjee. I'm a PMRF scholar in ISC, and I'm one of the teaching assistants of the course Host Pathogen Interaction. So, as you requested, we will discuss today regarding some assignments. So, week 7 assignments. The first the question is heat labile substances discovered by Jules Bordet that help in bacteriolysis are called option 1 antibody, option 2 complement, option 3 macrophages, option 4 defensins, option 5 histamines. So answer is the heat labile substance that is discovered by Jules Bordet that also help in bacteriolysis is called complement. Which antibody can activate the classical pathway most efficiently? So options are IgA, IgD, IgG, IgE, IgM. So our answer is IgM. Which of the following binds to the Fc of the antibody? C1q, C1r, C1s, C4b, C2a. So answer is C1q. Which of the following is equivalent C1q in lectin pathway? MBL, MSP1, MSP2, properdin, none. So the answer is MBL. In all except the following, CA is the smaller fragment. Option C2, C3, C4, C5, C6. Answer is C2. Which of the following acts as surface binding proteins and increase opsonization? Answer is C4b and C3b. Adaptive immune system. Different options are is germline encoded and can be inherited from parents. It's false. Has very limited potency. False. Has no memory. False. Is highly specific. So adaptive immune system is highly specific. Has limited or no memory. This is also false. Question number 8. Following protein is known to stabilize the C3 convertase. Option 1 C1q, option 2 factor H, option 3 properdin, option D C1 INH, option E proteinase. So the following protein which is known to stabilize the C3 convertase is called properdin. Which among the following statements is correct? Lectin complement pathway is initiated by binding lectin to mannose residues on glycoproteins. Lectin complement pathway uses C2 and C4. HIV cannot initiate lectin complement pathway. Answer is HIV cannot initiate lectin complement pathway. Which among the following is or are correctly matched? Factor B classical pathway, properdin alternative pathway. This is matched. Factor B lectin pathway. This is false. So the correctly matched is properdin alternative pathway. Week 9. First question is the quasi species in virus population are different viruses with same genetic makeup. Option 2 same viruses with same genetic makeup. Different viruses with some differences in genetic makeup, same viruses but some differences in genetic makeup. E viruses from same family. So the quasi species in virus population are same viruses but differences in genetic makeup. Which of the following is not a function of type 1 and type 3 interference? Option 1 killing of infected cells. Option B makes healthy cells resistant. Option C depress cellular synthesis pathway. Option D prepare and activate professional immune cells for development of virus specific immunity. Cleaves procaspase into active enzyme. So which of the following is not a function of type 1 and type 3 interference? Option 5 cleaves procaspase into active enzyme. 5' triphosphate single stranded RNA are sensed by RIG1. Before sensing, the ligand TLR3789 goes through proteolytic cleavage by enzyme cathepsin. MicroRNA, a type of non-coding RNA, can directly regulate virus replication at transcriptional level. First L that promotes apoptosis is present on TC cells. Which of the following virus proteins inhibit caspase-1 activity? Option 2 CRMA or cowpox. Antibody that masks surface proteins and stops virus host interaction and attachment is called neutralizing antibody. Which among the following statements is correct? Virulent virus is disadvantageous compared to the avirulent virus. Myxomatosis caused by myxoma virus in rats. Virulent virus kills the host rapidly, resulting in less time for natural transmission. Option C 1 and 3. Which among the following statements is correct? PRR sends viral PAM. Viral but not host DNA is present in the cytosol. Host but not viral nucleic acid are normally found in the endolysosome. Option 1 and 3 are correct. That PRR sends viral PAM and host but not viral nucleic acids are normally found in the endolysosome. So this is week 7 and week 8 content discussion. If you have assignment discussion, if you have any further doubt, we will discuss, or we will move into the innate immunity during virus infection part. Let me know if you have any doubt. So we will discuss about innate immunity during virus infection. So innate immune responses, pathogen recognition receptor we already discussed. So pathogen recognition receptor senses viral PAM. Direct detection of viruses by PRRs, viral signatures or PAMs are viral because viruses are replicating in host cells. Long double-stranded RNAs containing 5' triphosphate and unmethylated CpG motifs in viral DNA genomes. Additional factors help determines the viral origin. Viral but not host nucleic acids are normally found in the endolysosome, and viral but not host DNA is present in the cytosol. Indirect detection of viruses by pathogen recognition receptors, alteration in normal cellular processes such as acute decline in host protein synthesis, altered activity of ion channels, or ER stress not fully characterized. So, response to virus infection, innate antiviral mechanisms, type 1 and type 3 interference, pro inflammatory cytokines, adaptive immune responses, antiviral mechanisms, antigenic specific and long lasting antiviral immunity, cytotoxic lymphocytes, and natural killer cells, neutralizing antibodies. All these mechanisms ensures inhibition of viral replication, viral gene expression, and virion assembly. Innate immune response against viruses. Type 1 interference, interferon alpha, consists of 13 members and single IFN beta. Type 1 interference inducible genes, IP10, RANTES, etc. Type 3 interferon lambda, consists of 3 members. Inflammatory cytokines, interferon TNF alpha, IL6, etc. What antiviral factors are doing? So it can sense viral infections. So pattern recognition molecules, RIG1, MD, so RLR, RIG-like receptors, RIG1, MDA5, LGP2. These are adaptation complexes. NLRs, NOD1, NOD2, NLR C4, NLR P1, NLR 2 to 14. So sensing viral infections, adapted and complexes, NLRs, node 1, node 2, node NLR C4, NLR P1, NLR P2 to 14. So NLR P3 inflammasome, TLR, TLRs MYD88, TRI5, others M2P202. So these are cell intrinsic innate immune sensors, and these are cell extrinsic innate immune sensors. RIG1 MDA5 virus recognition pathways. So single stranded RNA containing 5 triphosphate. So RIG1 and MDA5 virus recognition pathway. So single stranded RNA containing 5 triphosphate, it detects, RIG1 detects. So and then long stretch of double stranded RNA detected by MDA5. So you can see after those get activated in the downstream, they keep on activating different adapter proteins. So Sendai virus, vesicular stomatitis virus, influenza, hepatitis C virus, Japanese encephalitis virus, small RNA encoded by DNA virus, Epstein-Barr virus, these are detected by this interferon, and then RIG1, and then long stretch of DNA are double stranded DNA is detected in the case of dengue virus, West Nile virus, recognized by both RIG1 and MDA5. Okay, so picorna viruses and can be activated by cytosolic synthetic double stranded viruses, poly polycytidilic acid, Laboratory of Genetics and Physiology 2. So MDA5 recognizes large double-stranded RNA structures lacking 2O methylation generated in the cytosol during virus infection. Sensing viral infections. So we have talked about this inflammasome activation by virus infection. So save influenza virus, DNA viruses, cellular stress such as disruption in membrane integrity and extracellular ATP released from stressed of damaged cells. So so there are different inflammasomes complex, you can see consist of NLRP3, AC, Pro-Caspase-1, NLRP3, and then M2, and another inflammasome complex. So all you know and this is NF kappa beta pathway. So all what happens is adapted molecules activates NF kappa beta pathway, and which produces pro interleukin 1 beta, and then pro interleukin 18 NLRP3, and then from this pro interleukin 1 beta it produces interleukin 1 beta, and this pro interleukin 18 produces interleukin 18, and it goes outside. Toll like receptors. So TLRs in mice, TLR 1 to 7, 9, 11 to 3, only 11 is functional. TLRs in humans, TLR 1 to 10, only 10 is functional. Cell surface, TLR 1, 4, 1, 2, 4, 5, 6, 11 and 12. Endolysosomes, TLR 3, 7, 8 and 9. Endosomal TLR trafficking and processing. So full length tailors, it gets cathepsins and its proteases, it gets cut, and then signal competent tailor are produced, tailor 3, 7 and 9. So mutations in these genes were found in two patients with severe HCV encephalitis. Endosomal tailor in virus recognition and signaling. Endosomal tailor in virus recognition and signaling. So TLR3 DNA viruses also produce double-stranded RNA by convergent transcription of their genomes. Dominant negative mutations in the TLR3 gene suffer from neonatal herpes infection. TLR7 human P disease, disease except CD8 plus disease and B cell census influenza. Mouse TLR8 expressed by myeloid disease and monocytes. TLR7 and 8 recognize single stranded RNA, uridine and ribose. The defining signatures of RNA are both necessary and sufficient for TLR7 stimulation. Viral fusion or uncoating and endosomal acidifications are required for TLR7 dependent recognition. TLR8 deficient mice develop autoimmunity due to hyperstimulation of TLR7. TLR, what is the function of TLR9? TLR9 is expressed in P disease and B cells in humans. Mice express TLR9 in P disease, B cells, macrophages, and conventional disease. Census viral DNA and bacterial DNA sequencing containing hypomethylated CpG motif. Recent studies showed that TLR9 recognizes the sugar-based backbone 2-deoxyribose and phosphodiester DNA irrespective of the CpG content. Type 1 interferon dependent antiviral pathway. So RNA interference, this is a process of RNA interference with the help of dicer microRNA gene. So this is nucleus where you can see that with this type 1 IFN dependent interferon pathway, how does it act? So ultimately, after microRNA gene or intron, it gets transcripted, and then pre-microRNA produced, primary microRNA, then pre-microRNA, then from pre-microRNA produce microRNA complexes and mature microRNA. Those microRNAs do mRNA target cleavage, translational repression, mRNA deadenylation. So RNA virus, viral RNA RIG1 from RIG1 mRNA, it produces RIG1, an inflammatory cytokine, and type 1 interferon induce antiviral responses, and host microRNAs, direct binding to MIR 485 with RIG1 mRNA, and degradation of RIG1 mRNA, then down regulation of RIG1 to maintain homeostasis, enhanced RNA virus replication. How doses of influenza virus. So So we have discussed the stabilized form of microRNA 324 can be used as antiviral therapeutic agent. The microRNA MIR485 targets host and influenza virus transcripts to regulate antiviral immunity and restrict viral replication. Type 1 interferon dependent antiviral defense, clearance of intracellular pathogens via autophagy followed by degradation in the lysosome is referred as xenophagy, uh, mm, and presents an important mechanisms of antiviral defense. Viral evasion of the innate immune system. All pathogens encode factors to counteract the induction, signaling, or antipathogen effector functions. Viruses employ distinct molecules to achieve blockade of the antiviral effect of type 1 interferons. This can be categorized into four major strategies: global inhibition of cellular gene expression, evasion from innate recognition, inhibition of molecules involved in interferon induction and signaling, inactivation of the interferon induced effector molecules. Cytoplasmic helicases, toll-like receptors. Mm, So toll-like receptors, these are different signaling pathways, and these are antiviral effectors, action of antiviral effectors. So antibody activities against free virus particles and virally infected cells. So neutralizing antibody, virus neutralizing cell protected from infection. Neutralizing antibody, so this is virus is infected cells. These are different neutralizing antibody which will act upon those viral spike like spike proteins and any other viral envelope proteins. So neutralizing antibody, this is anti-envelope. Non-neutralizing antibody, this is anti-NS1. This that doesn't neutralize. Non-neutralizing antibody, again anti envelope. So Fc mediated effector system, infected cell lysis or clearance, cell signaling, inhibition of viral replication, steric obstruction, inhibition of virus release, steric obstruction, inhibition of cell transmission. Effector activation of activated CD8 plus and CD4 plus T cells. CD8 plus T cell target cells, MSC class 1 plus. Effector mechanism, direct cytolysis, cell contact, granule, exocytes, perforin, granzyme. Facile T cell, Fas infected target cell interaction. CD4 T cell, Th1, MSC class 2 plus. Cytokine synthesis, IFN gamma, TNF alpha, chemokine. CD4 plus T cell, Th2. Cytokine synthesis, IL4, IL5, IL13. CD4 plus T cell, MAC class 2 plus. Cytokine synthesis, IL17A. Adaptive immune memory, memory B and T cells generated after infection or vaccinations are generally long lived. The factors controlling the formation and duration of the memory immune responses are not yet well understood, but several cytokines, B cell activating factor, may play an important role in maintaining the viability and basal proliferation of memory cells. B cell memory, long term humoral immunity. The following two principal mechanisms exist. Repeated exposure to antigen is one way in which memory B cell levels can be maintained. Pre-existing serum and tissue high affinity class-switched antibody to virus, probably from long-lived plasma cells in the bone marrow. Long-lived plasma cells secreting high affinity antibodies, long-lived, survive without cell division for many years in humans and contribute to the maintenance of antibody levels over decades. Antigen-dependent or bystander activation. Short-lived plasma cells survive for only a few days, produce antibody in extrafollicular foci, and probably crucial in very early response to pathogen. In Sweden, inactivated poliovirus showed substantial anti-poliovirus antibody titers in all age groups, suggesting the maintenance of antibody titers over decades in the absence of further vaccination or exposure to the live virus, but not true for tetanus and diphtheria. Antibody titers decline. Another study saw smallpox vaccination using vaccinia virus. Memory B cells show drop from peak responses occurred at vaccinations to an approximately 10-year time point. Memory B cells show a drop from peak responses occurred vaccinations to an approximately 10-year time point, but stable levels were seen between 10 and 60 years. Serum antibody responses could be identified at 60 to 75 years of vaccinations. It is possible that multiple mechanisms are involved in maintaining the serum antibody level. Humoral response to acute viral infection in humans. Systemic infections, chikungunya, Rift Valley fever, dengue. Chikungunya virus family, alpha viridae, persistence of an antibody 30 year, Rift Valley fever, Brunia viridi, dengue, yellow fever, measles, mumps, mucosal infections, coronavirus, influenza, RSV, rotavirus. T cell memory. Activated naive T cells give rise to both effector and memory cells. The relationship of the two naive T cell products is unclear. T effector cells higher frequency and their lower activation threshold to trigger cell proliferation and differentiation. Memory T cells can also rapidly express effector activity. Example: pro-inflammatory antiviral cytokines such as IFN G, TNF alpha. Naive T cell activation and proliferation and memory T cell population formation and maintenance are controlled by three cytokines: IL-2, IL-7, and IL-15. IL-15 dependent and long-term memory T cell viability. Suppression of apoptosis is supported by IL-7, which upregulates or sustains the expression of anti-apoptotic BCL2 gene family members in the developing memory T cells. Memory CD8 plus T cell differentiation. At the peak of CD8 plus T cell responses, naive T cell expansion leads to two distinct subsets: short-lived effector cells and memory precursor effector cells. This memory CD8 plus T cells further differentiate into self-renewing memory T cells, and the extended lifespan depends partly on IL-7 and IL-15 dependent homeostatic proliferation having show slow cell division and minimum cell number changes. So, adaptive immunization by viruses. Viral strategies to evade the adaptive immune response. So it can escape by mutation, it can escape by latency, escape by destruction of immune cells, escape by subverting antigen processing and presentation, inhibition of T cell mediated target cell lysis, inhibition of inflammatory response via modulation of cytokine actions, inhibition of human immunity by virally encoded Fc receptor, complement receptor, or control protein. Inhibition of class 1 or class 2 major histocompatibility complex synthesis. HIV TAT protein have been reported to suppress or inhibit MSC gene promoter activity. Human cytomegalovirus can impair MSC class 2 expression. Viral evasion of cellular immunity, escape by subverting antigen processing and presentation. So steps to interfere with MSC class 1 pathway: inhibit MSC class 1 synthesis, inhibit transporter associated with antigen processing, expression and function, inhibit MSC class 1 transporter, retain MSC class 1 in the ER, retain MSC class 1 in the pre-Golgi compartment, dislocate MSC class 1 to the cytoplasm, dislocate MSC class 1 to the lysosomes, bind to cell surface MSC class 1 molecules, increase endocytosis of MSC class 1 molecules. Virus encoded proteins: lentivirus, EVF, ABV, ACMV. Viral modulation of the cytokine system. Ways to interfere with cytokine functions: entrapped cytokine production, interfere with cytokine and chemokine synthesis, inhibition of generation of functional cytokines, interfere with cytokine actions, encode homologues of cytokines and cytokine receptors, type 1 interferon homologue, interferon gamma homologue, interleukin 8 homologue, interleukin 10 homologue, generally soluble cytokine receptors to neutralize cytokines, interferon gamma receptors, interleukin 1 beta R, TNFR homologue, interfere with cytokine effector function, alter cytokine signaling pathway. Cowpox virus cytokine response. Modified A, CRMA protein inhibits the production of caspase 1, which prevents the proteolytic cleavage of pro-interleukin 1b to mature interleukin 1b. Myxomavirus V, TNFR, and V, IFN-GR, or the vaccinia virus V, IFN-ABBP. Viral evasion of humoral immunity. Viral strategies to evade neutralizing antibody responses to the viral surface proteins include mutations of surface proteins by antigenic drift or shift and recombination, most notably of highly variable viruses such as influenza virus, HIV, and ACV. Masking of conserved epitopes, glycan shielding, as seen for HIV and Ebola viruses, for example. Surface protein decoy that may mop up neutralizing antibodies under certain conditions, for example, as with HBV. Original antigen screening, HIV and influenza virus. Irregular surface protein spacing that tends to reduce immunogenicity. Multiple surface proteins and virus forms, for example, pox viruses encode multiple proteins for entry. Virus induced immune dysregulation and autoimmune disease. Viral infections, particularly chronic viruses, are shown to enhance autoimmune disease in susceptible individuals, as infections frequently induce strong inflammatory responses in various organs. There are several major pathways through which viruses can initiate or more likely modulate autoimmunity. Viral infection, particularly chronic viruses, are shown to enhance autoimmune disease in susceptible individuals as infections frequently induces strong inflammatory responses in various organs. There are several major pathways through which viruses can initiate or more likely modulate autoimmunity. Direct infection of target cells or organs can cause the release of sequestered autoantigens and enhance antigen presentations. Local inflammation might alter the repertoire of self epitopes presented by APCs by altering antigen degradation properties of the proteasome. Presentation of pathogen epitopes with structural or sequential similarity to self epitopes might specifically activate autoreactive lymphocytes or molecular mimicry. Influenza virus and disease 1. So, Spanish flu pandemic 1918 influenza pandemic was the most severe pandemic in the recent history. It was also known as Spanish flu. It spread worldwide during 1918 to 1919. It is estimated that about 500 million or one third of the world's population became infected with the virus. The number of deaths was estimated to be at least 50 to 100 million worldwide. In India, the deaths were around 10 to 20 million. Mortality was high in people younger than 5 years old, 20 to 40 years old, and 65 years old. Emergence of influenza virus and pandemics. So, H3N8 Russian pandemic, H1N1 Spanish pandemic, influenza virus, circulation of H1N1, circulation of H2N2, circulation of H3N3. Who else can be infected by flu? Wild aquatic birds and natural hosts. Genome of IV, PB2, PB1, PA. So, genome of influenza virus. Influenza virus structure: negative sense single-stranded RNA virus, spherical to oversized range 90 to 100 nanometers, segmented genome 13.6 kilobase. This is virus assembly as uncoating, viral bird of attachment, internal framework, inhibit host machinery, and making copies. So this is the influenza virus structure, and then there different segments of their genomes, and these are associated RNA and then associated RNA polymerase. Negative strand single stranded RNA virus, spherical to ovoid range, rubber size, range 90 to 100 nanometer. This is influenza virus structure. Influenza virus disease. So 4 types of influenza viruses there. A type infect human and has pandemic potential, 8 segments of genome. B type infect human and has pandemic potential but cannot infect animal and can cause epidemics. C type mild illness to human and lack NA. D type infect pigs and cattle, no human infection. Naming of influenza viruses: the antigenic type A, B, C, D. The host origin examples: swine, equine, chicken, etc. For human origin viruses, no host of origin designation is given. Note the following examples. Duck examples: avian influenza A virus, A oblique duck oblique Alberta oblique 35 oblique 76. Human examples: seasonal influenza A. Geographical origin, strain number, year of collection. For influenza A virus, the hemagglutinin and neuraminidase antigen descriptions are provided in parentheses. Influenza A H1N1, influenza A H5N1. The 2009 pandemic virus was assigned distinct name A H1N1 pandemic 09 to distinguish it from the seasonal influenza viruses that circulated prior to the pandemic. So, how naming of influenza virus has happened? When humans are infected with influenza viruses that normally circulate in swine, these viruses are called variant viruses, and these are designated with the letter V. Example A, H3N2V. When viruses are isolated from non-living material, the nature of the material should be specified, like A, lake water, Wisconsin, I-79. Understanding the naming of a flu virus. A is virus type. This is place where virus is isolated, strain number of virus, year isolated, and virus subtype. Influenza virus and disease. Composition of influenza virus: RNA 1%, protein 73%, lipid 20%, carbohydrate 6%. The hemagglutinin receptor binding and fusion structure, fusion function. The major functions of the HA are receptor binding and fusion activities, but there may also be a structural role of the HA in budding and particle formation. The HA is a trimeric rod-shaped molecule. N is tetrameric, which is carboxy terminus inserted into the viral membrane, and the hydrophilic end projecting as a spike away from the viral surface. The overall amino acid sequence identity can be less than 50%. The structure and functions of these HAs are highly conserved. The cleavage size present in HA vary between viral strains and can affect tissue tropism, virus spread, and pathogenicity. The HAs of highly pathogenic avian influenza viruses of subtypes H5 and H7 contain a multi-basic cleavage site consensus sequence RXR-KR, which is cleaved by ubiquitous proteases such as furin or PC56, supporting systemic infection with an often fatal outcome. HAT, human airway trypsin-like protease, and TMPRSS2, transmembrane protein serine S1 member 2, also known as epitheliacin, and are present in human airway epithelial cells and have been shown to be capable of cleaving HAs having monobasic cleavage site. TMPRSS2 and the related proteases TMPRSS4 have been reported to cleave the HA of the 1918 H1N1 virus at monobasic cleavage motif. We can discuss more if you have any doubt. So this is the antigen antibody interaction mechanism. The interaction between antibody and antigen depends on four types of non-covalent forces: hydrogen bonds, in which a atom is shared between two electronegative atoms; ionic bonds between oppositely charged residues; hydrophobic interaction, in which water forces hydrophobic groups together; and van der Waals interaction between outer electron clouds of two or more atoms. In an aqueous environment, non-covalent interactions are extremely weak and depend upon close complementarity of the shapes of antigen antibody. So as we can as you had doubts, so we can discuss about the week 7 content again today as a revision. So host pathogen interaction, T cells and T cell mediated responses. T cell receptors. So antigen specific nature of T cell responses clearly implies the presence of T cell receptors. However, the T cell receptor is membrane bound and antigen recognition weaker than RCR. So this is antigen presenting cell, MHC class 1 T cell receptor, CD8. Self-MHC restriction experiment. This is T cell receptor viral peptide A, self-MHC. Two models were proposed: the dual receptor model, the altered self model. So two models were proposed: the dual receptor model, the altered shelf model. Structure of immunoglobulin and TCR. TCR CD3 complex. TCR accessory membrane molecule. This is CD4, CD8. TCR binding is weak compared to the antibodies. Roles of co-receptors in TCR binding affinity. So there are different co-receptors which helps in TCR binding affinity. These are also known as co-receptors. TCR multi-gene families in humans. So gene alpha chain, delta chain, beta chain, gamma chain, chromosome locations. Alpha B T cell receptor gene arrangement. Proliferation and differentiation, mucosal immune response, induce inflammation, antigenic specificity, B cell immunity, maintenance of cells tolerance, induce strong antibody response and allergic response, development of T cells in the mouse, positive and negative selection of thymocytes in the thymus, host virus interaction. So viral pathogenesis, changes in physiology resulting to deviation of vitals culminate to the clinical symptoms due to viral replication. Infection could be productive, abortive, latent, or reactivation. Viral pathogenesis pattern: acute infection, progressive infection, failure to clear all evidences of infections, latent infections, chronic or persistent infection. Chronic or persistent infections, persistence of any continuous replication, latency and reactivation, virus infection and disease outcomes, virus species and individual host, interplay of these factors, disease occur and disease severity. So, features of pathogens or virus: survive and multiply in the appropriate host. How it's possible: find a nutritionally compatible niche in the host, replicate using host resources, colonize the host, avoid, subvert, and circumvent the host innate and adaptive immunity. Strategies for pathogens and virus: not to kill the host. If it kills, then it should be highly infectious, known as facile transmission. Virus host co-evolution disease: myxomatosis caused by myxoma virus in rabbits. It's a benign disease in South America rabbit. So virus host co-evolution disease: myxomatosis caused by myxoma virus in rabbit. It's benign disease in South America rabbit. However, it causes 99.4 99.8% death. After a year, it causes 90% death. So subsequently, it causes 30% death, and remaining showed mild to moderate disease. The susceptibility of non-immune wild rabbits after successive epididymis and myxomatosis. Conclusion: rabbit developed resistance. Virus made changes to reduce the virulence, but it did not change to a virulent virus causing benign disease. Virulent virus is disadvantageous compared to the avirulent or resvirulent virus. Why? Because they kill the host rapidly, resulting less time for natural transmission.